I think we can applaud the fact that in a collective effort the time frame was more than kept. So thank you very much to all panelists also for this, because it allows us to have more time for debate. But I think it's only fair that I give the floor back to the panelists if there are questions, as we have seen so many interesting experiences demonstrated, whether the one or the other colleague might raise a question or needs additional information from our panelists. So if I can invite panelists, if there is a need to raise one or the other element for clarification, it's the time now. That seems not to be the case, unless you interrupt me. No, that's fine. This gives us even more time to give the floor back to you in the audience. And I see already the first demand for the floor. Mr. Hunzinger.
First, I wanted to express my thankfulness, my gratitude that you come here and discuss these questions. I really appreciate it that from so many countries, from so many continents, you take the effort to learn with us, because we see that this field is so young and is so fast progressing. We learn so much each year, and we are happy that you learn with us to really achieve this goal of this community to make a difference to the society and to the patients. Now, one thing we have seen this year in particular is the question of active targeting in combination with the topic of personalized medicine. Now, let's assume a hypothetical example. We have a lipidic or polymeric nanoparticle, which is well defined, which has been used in clinical trials and is on the market since 10 years or longer. Now, let's find out by personalization that this cancer nanomedicine may be more active if we can identify in a specific patient a specific receptor, which is specific for this tumor and may be unique for a patient. And we would like to introduce now a strategy of treating individual patients based on the individual receptor identified in their cancer. So we have a standardized nanomaterial, but we are able to exchange the ligand individually for each patient. Now, what we can expect is that this nanomaterial may be highly active, a microdose, total dose of drug. What we know is that there will be no animal model which is representative for whatever. What we know is that there will be no clinical cohort allowing us to do phase 1 or phase 2 trials to get statistics in these patients. Now, if this turns out to be a highly effective therapy for treatments with cancer, what will be the future way to regulate such a progress? On my side, the only way I see to do that is the way not to regulate the drug for this patient, because it will be too time-consuming and the patient will be dead long before the regulatory process may be finished. But the way what I could see is to regulate the process of manufacturing such individualized drugs. Now, is this something which fits into the current understanding of the regulatory process of nanomedicines? Or is this something which is just too different or to be considered?
Okay.
Thank you. I hope you are going to get many different answers, so I just put the first one on the table. We struggle with these questions because your final question of will that fit, the answer is we have to make it fit because this is the direction of travel, and of course what we want is highly efficacious and safe drugs, and if you manage to characterize very well that patient, which is not only going to benefit well, but very likely is going to suffer less side effects, that's where you want to go. So it's not only for nanomedicines. This is, of course, started with cancer, but other drugs and other products struggle with this issue as well. What we are finding, particularly in Europe, is, as you know, we are an agglomeration of many different national competence and national regulations. It's easier for us to fit the medicine than the companion diagnostic. So you first have to consider that not all diagnostic companions are going to be treated in the same way. Some countries may consider them as an inherent part of the medicine, and some countries may consider them that they are two different things, therefore you may have two different routes. That's our first problem. Now, the approach that we are taking is not to close doors for the future. The last thing that you want to do is to approve centrally a medicine in a way that in some country that medicine can be used, but the companion help to identify the patients cannot be fit into the system. So the way that we have to go is, even if the company has been developing the way to measure how that patient will be identified, we just want to put into the approval of that particular medicine that is to be used preferably in this. But we do not want to close the door that if that country doesn't approve the diagnostic, you cannot put the two together. Because as you very well mentioned, this is the future we will want to get. So it's many other things to put into the balance, but the approach has to be considered in advance so you can develop and improve and complement, but you're not closing doors with the approval of the medicine.
Are there different regulatory approaches to this question?
I'm not speaking because I disagree, to point that out first. Secondly, I would offer some options. One, I would say, use the opportunity to interact with the regulator early, because I cannot necessarily speak for all colleagues at FDA and CEDAR, and maybe if it's a combination product, to speak about, if you consider it a device in the drug, to address that. But early dialogue, discuss the science, consider things that we've learned about generics, and provide a strategy. I think there are also elements that can be considered that might be useful, such as developing and validating platform technologies that allow the plug-in of individual, in the context of personalized medicine, sequences or specific targets that are identified in a context. And if the platform technology has been developed and is, let me say, trustworthy because of pre-existing data or data existing from other mammalians at least, I think that's something to build upon. The question of companion diagnostic, of course, is critical after you have established some basic toxicological considerations. The critical issue often is how do you validate or qualify the companion diagnostic if you want to move it both forward at the same time. So I think there isn't necessarily at this point a single answer to it. Certainly the biomarkers and the correlates of effectiveness of a treatment, if there is evidence that you can establish a plan how to measure that in a relatively short time period, shifting the risk again into the later phase, but monitoring it very closely, I think it's another opportunity. And some of that we actually, at least for FDA, we do use in the context of the fast tracking and some of the, if it's a unique opportunity to address therapeutic benefit, I think that will fall into the equation and it will be considered. And again, without having very specific answers, I think early dialogue is very critical.
First, Mrs. Allen, number 2, number 3, if I understand correctly, but okay, number 4, for the time being. Mrs. Allen, please.
Yes, and I see the question as being slightly different. right now we have, let's say we have a drug platform, something like Doxil, which is an approved drug, which is even a generic drug now. We have had the technology for many years to, by a simple one-step incubation, to modify drug A, let's call it Doxil, to make it into an antibody-targeted Doxil by incubating it with HER2, by incubating it with Rituxan, which are approved antibodies. Is each of those to be considered now a separate and new drug that's going to have to go through a whole stage of testing, or is there any way, if we wanted to take drug A, which is an approved drug, and modify it with ligand A, B, or B, C, D, E, F, G, H, I, whatever, to be able to use those in a more personalized manner?
What do you think?
No, I mean, I think that it should, one should not have to go through a long and very extensive testing process to basically personalize an already approved platform drug, platform technology.
In principle, I agree with you. One should not go through excessive and expensive procedures to get there, but what you pointed out is essentially create a new entity by mixing two things together, and we
both of
discuss
which are
it.
approved probably already, like HER2 and Doxil, let's say.
Yes, but you know, you can discuss it with any clinical pharmacologist or any pharmacist for that matter. You're creating a new entity, and as such, the new entity needs to be considered, and you know, you have to establish the safety of it. And certainly it will behave differently in compartments, it will have a different PK, and so on. So the approach is you still have to go through the steps. And certainly it can be fast-tracked, and certainly there are ways to shorten the number of, or the amount of data that you may have to provide, because there might be, you know, there may be good correlates of clinical outcome, or there may be good ways to monitor the clinical behavior. I think that all can be discussed, and I think that that should be part of a dialogue.
I mean, and part of the same conversation is, how do you do a clinical trial when you have an N of 1 or an N of 5? I mean, I'm sure that's something that the agencies are already considering, but you know, we can't stall the whole system of by excessive regulation.
No one wants to have access of regulation, but you have to have enough safety data to go into a human. Right now, what is enough? Again, let's have a dialogue.
I just want to add that in case of medical devices, for example, we have a special access program, but it's being misused. Any new technology, any variation, the physician proves that it's the best one for their patient. Then you give them access to it. But then this becomes sort of a loop that it never gets to the manufacturing end process. And so one needs to establish some kind of reporting mechanism, so at least there's a reporting going on that will learn experiences that it's safe and effective. And over time you build some kind of data that proves that indeed this works in this group of patients. Even though you can't have a clinical trial done, you still have a means of accumulating information from experience. That's a program everybody values very much for devices, but unfortunately it's being misused, and chances are it might be changed because of that.
Anybody else on the regulatory side want to comment? If that's not the case, then the gentleman. So.
José Carballido from Basel, Switzerland. I agree with what has been said by the panel and by the participants. I think the most important is to start the dialogue as soon as possible. And I also don't expect that we will get an answer here. This is not the reason, but I think it's to start building maybe this interaction and to see how can we advance to have maybe solutions for things that they are very complex. I wanted to add also another example that I think is even more complicated, because now we could imagine that the drug doesn't exist. There is not a precedent. It's not a product for cancer that sometimes we have this kind of need that if we don't act very fast, the person may die. But we could think in autoimmune diseases, and we can think in active therapies. And in this case, it's even more complicated because what you have is a product that you will need maybe to deliver a substance, and that you will like even to target to a particular tissue or cell, and that, as it was said before, there is no animal model because this is all human-specific. And what we know is that there should be a rational and should be safety. But the safety will be very difficult because even you will not have even a non-human primate that you will be able to do it. And then what will be your first population? Because if you want to treat maybe a disease that you could imagine that you could provide a cure for people that they are having such a situation, but you are giving a vaccine, and you will not like to go even to a population that is healthy. First, because maybe it's absolutely irrelevant. They will have a particular MHC, and you will not be able to see any effect. But second is that it's a vaccine. And if you go to a population that is already advanced, so what will be the kind of overall recommendation? As I said, I don't expect an answer, but it's just to see the complexity of all the problems that we would like to use with this technology that brings really a lot of puzzles together and allowed us to make a big advance in medicine.
Thank you. Yeah, please, Mr.
I
Largo.
will take a partial access to that. The way that we deal, those are very specific cases, and they are a big mix of safety and efficacy. It has to be done one by one at the moment because we do not have a track record. So the group that we put together in this innovation task force is very multidisciplinary, because if the main issue could be a safety-related issue, you really need to see people who have seen a lot of medicines coming from the other side, not just this specialist on the really innovative part. And this is something to consider very, very strongly in this group, because we all know that this is a very young discipline, but more and more the way that the regulations of medicines is going is smaller groups, look ahead now, and plan how you are going to continue collecting information to be able to enlarge the group that will benefit from here. So when you are having specialists, for example, who are going to be proposing or asking you to think of risk management strategies and risk minimization, because you know your product or your idea very well, you may be able to say, okay, this may go this way or this way, but we can, from now, think how we are going to minimize the risk of stepping out of the pool of patients that we think are going to benefit. That could be a very wide range of measures. And then you may also be thinking how we are going to measure if our risk minimization are working or not. So again, if you are from the very beginning thinking of what is going to happen after we have an approved drug out there being tested by real patients, how are going to be our measures, our straitjacket, then you are going to have a much richer data to put on the table for the people who eventually have to take the benefit-risk decision.
Just as a comment, but maybe you have a chance further to elaborate, but then the question of Professor Allen comes back again, because if you would have to do this, as we have this specialized group in each regulatory system, the cost will explode again. And by that, the therapy or the product will not reach the market. So this is maybe an element. Frank, you would like to come in directly to that?
Yeah, I would offer some thoughts. I don't have all the answers, but at least let me pick one example. With flu vaccines, we have learned that, after the platform technology is established, you do a relatively small trial in healthy volunteers to just have some safety parameters of the particular combination of H and N that you put together for the particular season. And if you are not fast enough, the season is over, clearly, right? And we have many of those kind of situations that you just described, some of them. So I think we are in a situation where we need to rethink how we how we collect data, what kind of data do we collect. And one of the things that I mentioned is shifting the risk into either the post-marketing or actually into the treatment phase. But how do you do that? Well, I think many of you know that we have invested hundreds of millions of dollars into things on a chip, organs on a chip, a human on a chip. And that's all very good because it definitely helps non-clinical and preclinical development, and perhaps also is very useful for environmental sensor systems and monitoring our environment in a meaningful way. But it is clear that none of the chips represents just even a single individual, because it's some human tissue that is not reflecting human population, right? So there's significant risk. It doesn't do anything for clinical development. It doesn't shorten anything for the actual declaration from a product in clinical development, right? So it doesn't shorten cycle time at all. And as such, it was completely misrepresented. Instead, I would propose that we invest significantly in means to monitor real-time the well-being of human. And that will enable us to real-time also address risks with new and experimental treatments. And if you think of personalized medicine, it's the only way to go. But this is a significant investment. I mean, you all know what we measure today. I mean, we can measure some biochemistry relatively quickly, and we can measure heart rate and pulse and blood pressure, and we can run an EKG and an ECG and all of that. But that's not enough. We need to have a comprehensive battery to understand the status of a human well-being in a specific condition, and this specific condition will be an experimental treatment. So there's some work to do, and I think it's a stepwise process. But it's also a process of dialogue where all the stakeholders are involved, and where we understand that we are in a strategic alliance. And at the end we have one thing in mind, the benefit of the patient.
And of course it's how you judge the risk-benefit analysis in the end. Oh. The gentleman over there, if I understood correctly. No? Okay. Then Ms. Wolfram, then here.
Thank you for your presentations. Since we're talking about potentially setting up separate guidelines for nanoparticle approval, can you give an example of one toxic side effect that would be only specific for nanoparticles and not seen with any small molecule drug or biologics? Thank you.
Would like to take this? Maybe if you have an idea to whom you would like to ask, you could save time in detecting who would like to take it. Volunteers now?
Well, I can give you one example that's not even a nano drug, but it's a natural particle. It's silica dust, sand. If you inhale enough of it, you get silicosis. I don't know what specific example you're looking for, but certainly if you create, particularly if you make something new that we are haven't not been exposed to over time, and the organism, a human being or whatever organism, hasn't had time to either adapt or we haven't identified the challenges to it, we have to be very cautious. And I think that's really what the guidance documents say. There is no recipe to drive through, and there is no blank check. I think if you ask me, many of the guidance documents state nothing but to say, you know, in America there are those street signs. When you cross a bridge, you find the sign, slippery when wet. I mean, that's what we state in the guidance documents, right? Is that particularly useful? Not necessarily, in my personal opinion. What we have to do is be reasonable, be scientific, and deal with the facts.
Okay, then I have the gentleman here.
Yes, we have some nanomedicine product development happening in developing countries where the regulatory authorities don't necessarily have the capacity to assist in the development of the product or assessment of new chemical entities. So the question I have is, do your regulatory agencies have facilities to enable that early dialogue with nanomedicine developers outside of your countries? Say we want to approach the FDA and get assistance in developing the product.
Since maybe you mentioned FDA, I think I need to answer this. Well, I think there are different opportunities. We do have, as an organization, a outreach program for small companies that includes educational programs and information. Some of it is captured under the core competencies in regulatory science, for example. But as you can imagine, the agency also plans and has the desire and ambition to share all of its educational and training material with the world, because we believe that's a significant benefit, not just to the agency, but to humankind, to understand how things are regulated. It shouldn't be a black box. Specifically in terms of access to laboratory expertise, I think there are some groups that have in the past, let's say, decade specialized, and I look towards my colleague over there, the NCL, for example, one of the laboratories that has specialized for 10 years in characterization of nanomaterial with a battery of characterization methods and tests, I think could be a step into this direction. But I'm sure there are other opportunities in collaboration with, for example, centers of excellence, and perhaps even direct collaboration with the NCTR, National Center for Toxicology Research. So those are a few directions I would guide you. And I hope that I have captured what you're asking for.
Yes, additional comment from EMA.
Okay. Yeah, I'm looking for more of a direct early dialogue in terms of product development, like you mentioned, which is important for you to be able to commercialize the product. That's if there's an opportunity to have that direct conversation.
Okay, very briefly then, in Europe, the part of one medicine that you are ready for approval, there is a system, it's a specific Article 58, but this is at the approval stage, where you actually can use the European committees to get advice and to get a kind of we will approve this drug, even if the use is not going to be in Europe. But this is a later stage. I suspect you want the very early part. For the Innovation Task Force, we do not ask you where you live, so it's not close. Anything that is developed anywhere in the world may eventually be in Europe, so there are no barriers in that sense. You need to have well-defined questions that we can answer. If it's so wide that it's very early, we may not be able to help. But if we are able to help and it has enough material to judge, then where are you located is not really a limiting factor.
Ms. Lichemi? Okay.
While I really agree with the fact that preclinical and clinical trials are absolutely necessary, in the moment, what drives up the cost so tremendously is the fact that so many cell studies done in 2 dimensions do not really have high predictive value with respect to clinical outcomes. So on the other hand, we also know that there's a huge number of nanomaterials that we need to screen also in size-dependent and shape-dependent manners. And so I think what we need to address worldwide and with funding agencies is the fact that we need to develop culture models that go beyond 2D cell culture. First of all, we need to look very carefully in co-cultures of cells, whether that's now in an organ on a chip format where different co-cultures are coupled to each other. We need to understand much better how if you impact one cell type a little, how that impacts many other downstream physiological effects. There are also many new techniques out there that allow to go from 2D to 3D. So new efforts to grow microtissues, for example, where you then can control the build-up of the tissue, but also how built-up growth phases of tissue are maybe affected by nanoparticles. And I think we need to really ask, if we want to be effective in the regulation process. And we see we are not effective right now any longer. The costs are just too high. We need to ask in the moment intensively, what additional model systems do we need, and demonstrate for some of these intermediate model systems that they have higher clinical predictability than what we are currently using. And I think it would on a long term save so much money if we would invest into that now at an earlier stage.
Who feels tempted to answer? Or we take this as a statement? Okay, we take it as a statement.
Well, it is certainly a statement. I have Scott McNeil up there who may be tempted to address some of it, but meanwhile, while he's thinking about this, I would state that when I mentioned that the efforts in terms of the organs on a chip and the opportunity with such kind of model systems are non-clinical, and that it does not necessarily shorten the clinical development, it's not to criticize it as a very important tool to understand interactions of cells and even many organs, if you want, in their interactions. It's very valuable and it's very critical, and it even will perhaps propel and drive innovations in terms of new drugs and new materials and new opportunities. But one of the critical things is that it has been, perhaps by chance, misrepresented in the past as something that would speed up clinical development. It's not. Why? Because you stated it's not validated, and you have to go through you have to define the equivalency, you have to define a clinical correlate, and that's something has to be done step by step, just like with currently any other drug development tool or biomarker. There is the evidence that's needed. And without producing the evidence, you cannot jump into a human being. And again, coming back to shifting risk, if we want to justify to take the risk, to take something into a human being that hasn't been there before, and we haven't quite figured out all the impact in lengthy non-clinical animal systems that are quite expensive and still not predictive, then we have to do more in terms of sensitive monitoring systems, physiologic monitoring systems that are comprehensive enough to enable a physician to address at least the short-term toxic effects acutely, and certainly give a good idea about long-term effects, and then do clinical experimentation. But it has to be done in a mindful way, and it has to be done in a responsible way, and it also has to be done in a transparent way so that the public and the patient or the consumer can wade in. And that is one of the elements I think we need to make patients and consumers co-investigators, and they have to be part of the equation, otherwise we cannot solve the problem.
Thank you very much.
Then Professor Marcus, then the gentleman over here, then the lady here, and then the gentleman over there. And I'm afraid then we are close to 7 o'clock.
Thank you very much. So I would like to continue the idea which Patrick raised about personalizing medicine. And you imagine you are a doctor and you have a receptor or whatever, and you want to treat these patients, otherwise it dies in a couple of months. So I think we already have example for targeted cancer drugs. So when we make a sequence of the cancer genome, we can already select, based on the mechanism of action, targeted cancer that based on the sequence of the cancer. So it is unclear to me how the regulators want to develop not companion diagnostic but companion medicines to the diagnostic. And I don't even understand personally why is not approved if the mechanism of action is clearly a certain mutation, why not automatically approved what do the diagnostic company has to do more as a sequence, providing sequence data for that mutation, then to get a companion medicine. So what do you expect? Because it is unclear, I think, to us. It is not just a receptor or a sequence there, but this is not enough. You have to do additional studies, or what else do you need to do to get these targeted therapies to personalized medicine coming to the market? Because we already have that, many.
If I would be the only regulator at the FDA, I would offer an approach, but I appreciate the passion, and I know that it's a challenge, I think, for all of us who are concerned to bring medicines to the patients faster. I think some of the elements to consider are certainly traditions in the whole process, the level of exchange of information. So when we talk about education, that includes also FDA staff and regulators across the globe, I believe. And yet there are still challenges. If you make something and you put it in a person, there has to be some reasonable evidence that it is something reasonable to do. So if you create a new entity, in a sense, by mixing it together in a specific individualized format, even if it's on a platform technology, and coming back to the example with flu vaccines, may somewhat educate the kind of point I'm trying to make. You cannot just make a new vaccine and distribute it broadly. I mean, there has to be at least some level of safety that you build into it. But again, let's focus on the dialogue. Let's point out the weaknesses in the current approach, and certainly there are things to do in terms of education for not just the public, academia, industry, but also certainly education and training at the regulatory agencies.
Okay, I want to come back to several. I'm here. He was looking. I'm here. Because you're the main, you know. I want to come back to 2 points and do it very shortly. First of all, to the clinical trials, which, from my point of view, coincides also with the cell on chip. Because, you know, the effort that has to be done are not only in it was mentioned in several of the lectures in this meeting, which I think is very important. We have to develop new tools, both for sophisticated clinical trials, especially if we want to go to the personalized medicine. The clinical trials has to do nowadays a big jump to adjust to small sets, to specific issue, and also, like you said, to the big picture regarding the cell on chip. The cells are just part. We have to go all the way to relate you know, to the whole human being, even to the climate. And therefore, you know, the FDA that gives the regulators and all the countries in the world are looking towards that, sometimes it's not right for the different weather or different culture or different other things that we relate, and it is also relevant to nanomaterials. It's not excluded. So I think we need to invest, as you said, regarding the invest on cell on chip, on new tools, on new analysis, on a new approach, in parallel to the broad picture that we need to understand. Thank you.
Well, in that context, I can tell you that particularly in the past 6, 7, or 8 years, FDA has made significant progress towards making the agency a science-based agency, and all of those who have been following it, you know that we have engaged into a network of scientists and reviewers in having access to international experts in our review process, for one, we have built centers of excellence with academic institutions in the United States, but that's not limited to the United States. So any additional boost in financing, because we do have to seed fund some of the centers, even though they are in many ways one of my goals to the centers that we engage in is to make them self-sustained. And I think that's easily possible because many stakeholders understand the importance of this relationship of scientific excellence and interaction. We also use another mechanism at FDA to engage into research and problem solving. It's a very nimble contractual mechanism. It's called the broad agency announcement. It doesn't really describe what it is, but it's a very nimble contract mechanism where we can engage with anyone who either we believe has the capacity to solve a specific problem, or where applicants come to the agency under the priorities of regulatory scientific challenges and offer new solutions in a contractual relationship. And that program has been going up very steeply. So it's definitely working well. It's both a grassroots problem addressing mechanism for the centers, but it's also a horizon scanning mechanism for whatever the latest technologies or solutions might be to address regulatory science questions. And I think we've come quite a way. The other part that I mentioned, in any of the centers of excellence and also in other interactions, we do emphasize the importance of education, not just the education in academia to make future regulators and scientists knowledgeable of the complexity of a regulatory process or even product development in itself, but also to train FDA staffers and give the opportunity for scientific exchange and stimulate the scientific excellence within the agency. So I think there are some programs, just to mention a few, that support this goal.
Just to complement a little bit, an agreement with all has been said around this area, but you also have to remember that when we are talking about these early dialogues, the methods and how you gather is a keystone. So it's very good to think of the facts and the evidence, but how you are going to arrive to that evidence is part of the discussion, very important, especially at the earlier stages. Again, information and communication has all the dimensions that have been mentioned, and then add another one, which is we have to think of the patient. So how is this new treatment diagnosis approach going to fit in the health system? Because when that element is put earlier on, you actually get a different color of which direction to take, which can be very, very helpful.
Sure, yeah. Thank you. So far the discussion has been mainly on medicinal products. I'd like to briefly give some information on the situation for medical devices. I think it might be relevant also for people here. In Europe, the current regulatory system for medical devices does not have any provisions for nanotechnology or nanomaterials. However, that regulatory system is in revision, and in the proposal that is currently still under negotiation, both in the Council Working Group and in the European Parliament, there are some provisions for nanomaterials. So we don't know what's going to be in the final text, but there are some provisions. Furthermore, there is a European Commission working group on new and emerging technologies in medical devices, which tries to gather information which would be useful to get the situation better in relation to the provisions that are proposed in the regulatory proposal. Furthermore, there is an ISO TC 194 on biological and clinical evaluation of medical devices that is currently developing a guidance documents for nanomaterials. In the group are a lot of industry experts, but also representatives from governments of the Netherlands and of Canada, of the US, of China, and of Japan. And I think we're making really good progress on that document. We are hoping to finalize that within the next one or almost 1.5 to 2 years. And finally, people may be aware that also the Scientific Committee on Emerging and Newly Identified Health Risks from the Commission is working on a mandate for providing guidance on a risk assessment of nanomaterials in medical devices, which is expected to be coming out at a relatively short notice as well. If there are people that want to know more about these developments, I'm open for questions, of course. I'll be here also this evening and tomorrow. Sorry.
Maybe very briefly, thank you very much. I just want to remind the audience, when things are moving as they are at the moment in this field, you really need to make an effort to comment when the public consultation came, because this is the time to really put the important issues on the table and make people react to them.
That's a very valid comment. Now you, in the center.
I would have a question accordingly. If we have a tissue-engineered product where the scaffold is produced using nanotechnology, to what extent should the cellular component be included in the risk and safety assessment of the whole product? Per ATMP regulation, the whole product, scaffold and cells, should be considered. How should we extrapolate this to nanotechnology-based scaffolding or constructs?
It might be considered a combination product, and we look both under devices and drugs and biologics for that matter. And I know some products are already coming in for review, but this is when you have to contact a regulator, because it's difficult to address every angle of these implications. These are new way of approaching these devices. These are hybrid products almost that we haven't had before. And our regulations are old. They don't necessarily account for these novelties. So we have to come up with guidances, policies that are allowing to look at those devices and products that are coming to the market. Our regulations are ancient. So when a new product comes in and you're bound by the regulations, it makes it impossible to do anything outside that regulation. So it is best to actually open that dialogue that we're all talking about, which would probably put some pressure on regulators to revise and review and maybe have a whole new set of regulations specific to these products that we're going to see in the near future.
My safety concern would be that the the cells are affected by the interaction, and due to this interaction, a safety issue may arise due to application of the combination product.
Probably, I'm sure, but I don't know how one would go about assessing both components and in combination, and also where it's going to be applied probably would be also impacting on the decision-making process. It's like an artificial organ implantation.
Yeah, in support of that, I would say it's certainly a question as a combination product. What do regulators do? I mean, the example of the FDA, we recognize the challenge. We created an office under the Office of the Commissioner that specifically deals with combination products, and of course also coordinates the interactions between different centers. As you may remember from my introductory slide, the centers itself have had in the past, they had to function very independently. So they created their own structures to regulate. And as such, sometimes there is what we internally call the silo effect. But the goal of this office to coordinate also across specific disease groups and areas is critical for facing up to the future and address that.
Over there, then Mr. Weltering, and okay, then you, but then I would like to close the list. We have started later, so we can easily accept to be late now too, but I don't want to bring the organizers into problems by taking too much time. I get a signal it's possible to go further, so if there's even more interest to participate, I have noted the gentleman over there was first.
Okay, one thing about what Frank was saying about the man on chip, whatever, and tissue culture and all this. I think the point is that with the nano drug or nanomedicines, whatever, most of the effect is dependent on the ability of this nanoparticulate to control biodistribution in a very clever way. And unfortunately, we cannot achieve this on any of the tissue culture or even 3D systems because of the microanatomy that is so complicated of these tissues. And actually, I just want to say one thing. If we would deal with, let's say, development of a nanodrug, from my experience, when we developed Doxil, if we would take the tissue culture data that we have and use them, we would not develop Doxil because the stuff doesn't work. So I think you have to be very, very careful. If sometimes you can kill a project for a wrong reason if you use the wrong system to evaluate it. You have to be very careful. So I think we still need to find what will be maybe a simpler system than the whole animal or all human being, but at this point of time, I don't think we have replacement to them, as you said. I mean, and the value of the other system are, from my perspective, sometimes the opposite. Namely, now I know that if I want to develop a good nanodrug that will not release the drug immediately, I'll test it in a tissue culture. If it's not active, I'll say, well, it doesn't release the drug, so it may serve the purpose. But otherwise, it's very limited as it stands now.
I agree. It's a complex system. I'm not sure it's a question in there other than sharing your experience, which we probably all agree with.
Yes, since we have all these regulators lined up so nicely, I thought I can ask a very general question. I have the feeling that the gap between technology, the speed of technology development, and the, as you stated, the ancient regulatory processes becomes bigger and bigger. So the question now is, how do we cope with this? Is dialogue just enough to cope with this? I mean, how can we speed up regulation to keep pace with the technology development and the speed of that, which is not applying just to nanotechnology. It's in general, I have the feeling that since if you think about 3D printing, for example, I mean, progress is so fast. So how do regulators cope with this speed? And maybe everybody can answer.
Okay, I'll start, but I think this is an interesting one to go down the line if you want to. First, it is maybe true. I'm not sure about the size of the gap, but the order is right. The regulators have to go behind the times. If we put the horse in front of the cart and all these things, that will not work. So we are happy with the system. Now, it is important to watch that gap and make sure that that doesn't go any further. You have to remember that regulators are risk adverse because when we are talking about new things, everybody is happy to, the earlier to the patient, the better. But then everybody will be extremely unhappy with one vaccine that goes wrong. And I'm going with 2 vaccines because they have been mentioned and it's the obvious one to blame here because you start with healthy patients. So I agree that watching the gap is very important. And for us to be here in early time, good, enthusiastic new field is one of the way to do it. So dialogue is, for me, the main part. The second is also to be very aware that we are putting another layer now, which is the cost conscious of all these national competent authorities who eventually will have to think about what they do with the drugs. We cannot ignore that. So yes, we need to always assume that the gap is going to be there and to make sure that we are in an elastic way, never let it too much, but never put it too close. So good conferences, well-organized dialogue, and also taking the education outside of this room to the clinicians and to the patients, as had been mentioned before.
Usually, if regulations are putting limitations on you, you end up looking for expert advice. What is the best way to go about the product? Is it ready to be out there? Is it safe enough? So if there's a clinical opinion out there based on their experience. We are ready to hear what they have to say, and at the end of the day, of course, as a regulator, we are going to make a decision, but we're looking at every possible resources. When it comes to putting new regulations forward, there's a lot of steps we have to go through to prove that the only solution is a new regulation. And if that be the case, then eventually we are going to have new regulations like for tissues and organs. But for now, we feel that existing ones have enough room that with additional guidance and policy, we would be able to cover with new standards and methodologies that are being developed and hopefully will be, proven to work and be repetitive and consistent. Now we don't even have methods we can tell the manufacturer this is okay for us. They have to use several methods.
Well, first of all, let me clarify that FDA regulations are not ancient, all right? Most of the stuff just came out after 62. I think some of it has to do also with human nature, right? I mean, the majority of human beings are conservative. They shy away from new, from change, and so on. And that's probably part of the answer. But that's not an excuse. So I don't want to make an excuse why we are having often a gap between technologies that approach the agency or questions that society has towards regulatory processes. But I think there are ways to make the gap smaller and build dialogue. And we have to make sure we get the resources to have excellence in science also at regulatory agencies, because I believe that science is the underpinning element of regulating and making policies.
Well, the question is if you really are willing to adapt these new inventions. For example, if you have contamination of food, it's still required that you do these plate tests, which take 3 days. But the customer gets the result with PCR within 2 hours. So the question is, these kind of adaptation of methods is also a question.
You're absolutely right. We change actually the food surveillance process towards newer methods, but you also can imagine that US-wide, I forgot how many lines of imports we have, but it's in the millions. Right, and certainly there are field offices that you have to harmonize as well, because you cannot have a field office measure something different from the other. There's laboratory information systems that have to be standardized and updated and so on. So it's not always done overnight, but I can assure you, and if you want to look up in the context of the CIFSA, Center for Food and Nutrition, Advanced Nutrition, the scientific program and the goals in the context of the Office of Regulatory Affairs, ORA, which basically are the boots on the ground to implement and do that, I think you will see a lot of change that is actually happening.
If you allow me, as I represent, let's call the political level, I think one has to be clear. There are two elements. In a democracy, legislation or regulation is always slower than innovation. The point is whether systems are in place allowing to react in time, and to make this possible, it is needed, and here I have to echo what was mentioned by most all regulators, is that those being innovative have to share via discussion, via information, at a much earlier stage, as normally they might be tempted to do so, this information to enable regulators to put the mechanisms which have to be provided in place and to activate them. And secondly, you have to do this because don't expect in these days that politicians are able to understand you easily. So, and this is not a criticism to politicians, because they are part of a society which does not consist only of engineers or natural scientists. So that we have to accept whether we like it or not. So I have now the gentleman here, then Mr. Loeffler and Bruce over there.
We all complain about the costs and the extended time, which makes it very expensive, maybe exploding costs. My question is, how do you think, would it be ideal maybe to think of having a central world agency in 2020, or do we better cope in the long run with the diverse agencies?
I'm afraid we have to be realistic.
I think...
I have a question.
Yeah.
It was the German industry. Sorry? It was only the German industry complaining. The French had it already.
You know another story. I have a question. Now comes the question. Do the regulators have a mind map on what their needs are and how much expertise characterization they need? Maybe they should, by politicians and by regulators, say, this is our need list, and probably innovative industry would respond to it.
Yes, I think that's a good approach, and FDA actually has communicated and published the priority areas in terms of regulatory critical issues and questions in regulatory science. There are 9 or 10 priorities at this point, and you can find them on the web, and they are defined to some of the detail that you can get a good idea where the shortcomings are. So I think we try to communicate that in real time as much as possible. We renew the announcement, for example, for this BAA process that I mentioned for the contractual interactions on a at least annual basis, but as soon as there are new questions coming up, we go and change the questions. So that's one format. The other format is, of course, interaction with the public and politicians to point out what are the critical issues and where are we struggling. And that's a complicated process, as you can imagine, particularly with the government where the Congress or in a sense, the Parliament doesn't work with the government, and as such, it's a very difficult situation to get resources reassigned. But the signals from the current government in the US is very positive towards addressing those questions, being close to the speeding up science and education, and closing the gap between regulators and even, let's say, agencies, government agencies that fund innovation. I mean, if you think about the principle in US about funding, many of the products that we are seeing at the regulatory agency, probably 50% of those were initially funded in its initial onset as a research project, again, by one of the government agencies. So you have some expertise in other government agencies that we can benefit from in a dialogue and networking, and that's exactly what we are doing. The problem is that there isn't really an additional resource to make the coordination happen, so it's on the existing budget, and there is a little bit of human gravity that and sometimes it's gravity of the mind that works in the process. But I think the goals are quite well defined. I don't think we lag behind there. I think we need more and better solutions.
Because the noise from outside is getting more and more louder. Professor Bruce.
I'm not actually a professor yet, but anyway, doctor only. Partly in response to Klaus's question, which I have the same concerns, when Dolly the sheep was cloned in Edinburgh, I was much involved with the ethical questions, but also a little bit legal questions. And a friend of mine who is a medical lawyer said, I'm not sure this is covered under the Human Fertilization and Embryology Act. And I phoned up the Human Embryology Authority and said, what are you going to do about this if it's not covered? And they said, you don't realize how much effort is required to do primary legislation. It takes several years. We will try and deal with this in the context of the existing Act. And that seemed to be okay. And then there was a legal challenge later in the year to the question of whether it was covered. And this was, and it found it wasn't covered. And so the thing went through a whole judicial review process. And then the mad Italian scientist claimed he was now going to start cloning human beings. And suddenly, there was no legislation to stop it. He was going to come to Britain because Britain had no effective law. They passed an act in a week. So it was interesting when there was a political will to do something, people could move very fast. So it was just an illustration that there was genuinely a situation where this was totally new and it was not covered in legislation. It turned out, logically, had it been there, the Act would have covered it. So they went through a high enough process and eventually they sorted it out. But what interested me was how fast they could move when they had to. I just wonder if that's an abnormal situation, or is that something which could also happen in USA, for example?
Well, if I may comment, since you mentioned the USA, I predict that the legal framework for data and personal data will change. It has to change because otherwise we cannot really do personalized medicine. And that's in the context of data mining, in the context of social and behavior science and data that has to be collected. And in a way, if you think of those nice little things that we all carry around, with a little bit more sophistication, they actually can paint a very good picture of our own well-being and unwell-being in a relatively simple way. And we have to learn, and we have to find a legal framework that allows for taking the benefit as a society and as an individual from such kind of information. So I think there will be change, and it will be rather soon, and sooner than we actually think the government would move on it.
I think it would be only fair, before we close this afternoon session, that I give the floor to all panelists for a final statement, maybe with one exception, because you have been addressed at several occasions. So let's do it the other way around.
Well, after all these questions, I just wanted to say, as has been said, of course innovation comes first and then we come behind, and we also have to learn about the new technologies and the new challenges, the new risks that the technologies pose as nanomedicines, as we have seen in the conference during these days. But I think, as has also been said, if companies start an early dialogue, we are really open for it, and we try to understand with them what the challenges are and to implement it in the evaluation of the dossiers. So, yeah, please come early and discuss.
Thank you. You're not obliged to take a last word, but if you want, no?
You want to hear more about the lizard?
Yeah.
This technology innovation and regulatory issues, not only in nanomedicine, in other areas also, they are neck to neck. Sometimes regulatory issues will be delayed, but ethical issues are very important. Here, somebody lives for 40 years, somebody lives 80 years. And that 40 years person who is dying at the age of 40, he has every right to live up to 80 years. So ethically, we have to think this also. When we frame a regulation or anything, how much benefit patient is getting, what is the cost effectiveness to the benefit, and that if we have compromising on both ways, then only the science like nanomedicine will come in the for patient care very, very future. Thank you.
Yeah, I'd just like to reiterate that the regulation always seems to follow the scientific developments, and these regulations, I don't think it's unique to nanomedicines. I think there are other areas of any technological development we need to keep abreast. I think at the TGA we've had a reorganisation which has slowed us down a little bit, but I'm really very keen to get together with the other regulators tomorrow and continue the dialogue.
I've said enough, but I don't think that regulators only trail innovation. I think regulators do actually innovate in the context of addressing issues that no one else sees and reaches out. We do reach out to address problems, and one of the mechanisms I did describe, but that's just for the record.
Just one sentence. Personally, I believe nanomedicine is destructive. So I think in the near future, all of the medicine are personalized. So as long as efficacy is overweight potential safety issue, I think we should go ahead. Probably the current landscape will be changed. That's my personal view.
Well, we do have open dialogue with companies, with associations, and that's another way of them bringing these issues forward. And when it's a voice that comes with group behind it, I think they listen and they pay attention more. This is something that is starting to make an impact, and regulators are starting to think about it in ways, how are we going to regulate these products now that they are here? Until now it was almost like a science fiction project, and now we can see endless applications of it, and we have to face the reality and say what is the best way about going, assessing some of these that are going to be one of a kind type of products. And hopefully there will be more attention and more resources, which is right now at minimum probably that is required to work on these type of projects.
So I think that the regulator has accumulated very knowledge, and also we have to accumulate the consensus between the regulators and the industry and the academias. So from the regulatory side, I think that the reviewers and regulatory researchers and policymakers has exchange opinions and make cooperations to shorten the gaps between the industry or academia and the regulators. Thank you.
Not to repeat, just to stress that what you do is extremely important, so keep innovating and keep giving us feedback to make the wheel go faster.
So let's, with these final words, conclude our afternoon session. I would like to thank you for your patience, but I think as the discussion has demonstrated, there was a certain need to make use of the accumulated knowledge and the accumulated presence of regulators in this room. I would like to praise the organizer for having put for tomorrow on the agenda session number 33, where I strongly believe that one of the main issues to be discussed tomorrow needs to be how regulators organize the cooperation in future in more, let's say, formal terms, because what became clear this afternoon is if we don't do so, we will block innovation because of the costs involved, and if not regulators work together and share the information related to personalized or more and more personalized medicines, we will not be able to serve the patient's needs. And this is probably the need to be discussed tomorrow morning. Thank you very much to all of you.
May I